This is week 7, Complex Inheritance Patterns, part 1. The learning objectives for this week are to describe the basis of multifactorial and polygenic inheritance, to use empiric data to determine risks to individuals and family members, and to give examples of birth defects and common adult conditions with complex inheritance. Few genes act alone. Environmental factors and other genes may modify expression. Traits can be described as being either Mendelian, caused by single gene, or polygenic, caused by multiple genes. Both can be multifactorial or complex due to an interaction between the genes and the environment. When you look at the genes, the environment, and the traits, we see that single gene traits are discrete or qualitative. They often produce an all or none effect. The polygenic traits, though, produce a continuously varying phenotype, also called quantitative traits. The DNA sequences involved are termed quantitative trait loci, or QDLs. Some examples of polygenic traits are height, skin color, body weight, fingerprint patterns, behavioral traits, congenital heart defects such as tetralogy of Fallot, a ventricular septal defect, a VSD, an atrial septal defect, an ASD, and a PDA, a patent ductus arteriosus, and neural tube defects such as anencephalia and spina bifida, as well as cleft lip. We need to remember in the case of the polygenic traits that the individual genes follow Mendel's laws, but their expression is hard to predict. And the effect of genes is additive or synergistic, and the input of genes is not necessarily identical. The frequency of distribution of the phenotypes forms a bell-shaped curve. Dermatoglyphics is a study of fingerprints. The number of ridges is largely determined by genes and prenatal content. The skin on the fingertip is folded into these raised patterns, called dermal ridges, that align to form loops, whorls, and arches. A technique called dermatoglyphics, which means skin writing, compares the number of ridges that comprise these patterns to identify and distinguish individuals. The average total ridge count is 145 in a male and 126 in a female. This slide is showing you the anatomy of a fingerprint. It shows the total ridge counts for a number of individuals plotted on a bar graph, and they form approximately a bell-shaped curve. The number of ridges between landmark points A and B on this loop pattern is 12. You can see in the inserted diagram. The total ridge count includes the number of ridges on all fingers. It's important to note that dermatoglyphics is correlated with genetic abnormalities, and it's useful in diagnosis of these disorders at birth or soon after. Abnormal ridge patterns are seen in the following syndromes: Down, Pateau, Edward, Turner, and Klinefelter, and they're seen in some other disorders too. It's also important to remember that forensic fingerprint analysis is an application of dermatoglyphics. The inheritance of height. Genetics students at the University of Notre Dame lined up by height in inches, revealing the continuously varying nature of height. The effect of the environment on height is obvious. People who do not eat enough do not reach their genetic potential for height. If we were to line people up according to height, but raised in two different decades and under different circumstances, this could illustrate the effects of genes and the environment on this continuously varying trait. Students from the 1920s, on average, were considerably shorter than students from recent years. The tallest individual from 1920s were around 5'9", and whereas the tallest today are about 6'5". The difference in heights between then and now is attributed to improved diet and better overall health, and perhaps the fact that many men were killed in the First World War in the years before the first heights were measured. At least 50 genes affect the height. And certain SNP patterns are seen in individuals with periods of rapid height increase, and we will discuss that in just a moment. In looking at skin color, melanin protects against the DNA damage from UV radiation, and it's actually exposure to the sun that increases melanin synthesis. Humans have the same number of melanocytes per unit area of skin, but they differ in the melanosome number, size, and density distribution, as we discussed when we were discussing discussing albinism. If we look, we see that there are more than 100 genes that affect pigmentation in skin, hair, and irises of the eye. The definition of races is actually based largely on skin color, and it's a social construct more than a biological concept. Skin color is not a reliable indicator of ancestry, and 93% of varying inherited traits are no more common in people of one skin color than in any other. In order to investigate and predict the chance that a polygenic multifactorial trait will occur in a particular individual, geneticists use empiric risk, which is based on the incidence in a specific population. Incidence is the rate at which a certain event occurs, such as the number of new cases of a disease diagnosed per year in a population of a known size. And prevalence is the proportion or number of individuals in a population who have a particular disease at a specific time, such as during 1 year. Let's look at cleft lip. Cleft lip is more likely to occur in a person who has a relative with a condition. And what it is one or more fissures that form in the upper lip. Folic acid has been shown to decrease the incidence of cleft lip if it is taken during early pregnancy. It has also been shown to decrease the number or incidence of individuals born with neural tube defects such as anencephalia and spina bifida. Empiric risk is not a calculation. It is a population statistic that's based on observation. Therefore, if a trait has an inherited component, then it makes sense that the closer the relationship between two individuals, one of whom has the trait, the greater the probability that the second individual has the trait too, because they share more genes. If you look at this table summarizes empiric risks for relatives of individuals with cleft lip. Here we have identical twins, 40%, a sibling, 4.1%. And if we go down to the general population with no affected relatives, we give it a 0.1%. This slide shows heritability estimates the genetic contribution to the variability of a trait, and observed variance in a multifactorial polygenic trait or illness reflects genetic and environmental contributions. Heritability equals 1.0 for a trait whose variability is completely the result of gene action, such as a population of laboratory mice who share the same environment. Without environmental variability, genetic difference alone determines the expression of the trait in a population. Variability of most traits, however, is due to differences among genes and environmental components. If you look at this table lists some traits and their heritabilities, such as clubfoot, height, mathematical aptitude. Researchers use several statistical methods to estimate heritability. One way is to compare the proportion of people sharing a trait to the proportion predicted to share the trait. The expected proportion is derived by knowing the blood relationships of the individuals. This slide is showing the measurement coefficient of relatedness, which is the proportion of genes that two people related in a certain way share. And you can see sibling to sibling, parent to child. Both of those are a first degree, and their percent that they would share would be 50%. In second degree relatives, you have 25%, and in third degree relatives, it is 12.5%. If we look at this slide, which is basically tracing relatives, we could look at the arrow which is pointing to the proband, which is Tim, the index case. Tim has an inherited illness. A genetic counselor drew this pedigree to explain the approximate percentage of genes that Tim shares with relatives. This information can be used to alert certain relatives to their risks. Please go back and look at the coefficient of relatedness for pairs of relatives, and you can see the primary, secondary, and tertiary degrees of relationship, first degree, second degree, and third degree, and you can relate it to this pedigree. An adopted person typically shares environmental influences, but not many gene variants with the adopted family. Conversely, adopted individuals share genes, but not the exact environment with their biological parents. Therefore, similarities between adopted people and adoptive parents reflect mostly environmental influences, whereas similarities between adoptees and their biological parents reflect mostly genetic influences. Information on both sets of parents can indicate how heredity and the environment contribute to a trait. Studies that use twins to separate the genetic from the environmental contribution to a phenotype provide more meaningful information than studies of adopted individuals. The term concordance measures the frequency of expression of a trait in both members of monozygotic or dizygotic twins, and twins who differ in a trait are said to be discordant. For traits largely determined by genes, concordance is higher for monozygotic than for dizygotic twins. Remember, the monozygotic twins came from the same egg. The dizygotic came from two different eggs. This table shows concordance values for some traits in twins in comparing monozygotic twins and dizygotic twins. Let's remember that diseases caused by single genes approach 100% penetrance, whether dominant or recessive, and also approach 100% concordance in monozygotic twins. That is, if one identical twin has the disease, so does the other. However, among dizygotic twins, concordance generally is 50% for a dominant trait and 25% for a recessive trait, since they are basically siblings. These are the Mendelian values that apply to any two non-twin siblings. For a polygenic trait with little environmental input, concordance values for monozygotic twins are significantly greater than that for the dizygotic twins. A trait molded mostly by the environment exhibits similar concordance values for both types of twins. This slide just shows you the differences between dizygotic twins, monozygotic twins, twins raised apart, and adopted individuals to sum up from what we were discussing.